Hi everyone. In this video, we are going to be talking about the genetics of blood typing. So I don't know how many of you all have donated blood before, or if you know what your blood type is. But if you have donated blood, then perhaps you know that that blood can only go to certain groups of people depending on what the recipient's blood type is. So if you're a type O blood, people try to get you to donate that blood a lot because, as you can see from this chart, type O blood is able to be given to anyone, regardless of their blood type. But on the opposite side of the spectrum, if you are type AB blood, like myself, that AB blood can only be given to other AB people. So it's a lot more restricted in terms of who it can go to. And then A and B are sort of in between. So why is this the case? Why is there this difference in terms of who can give blood and who can get blood depending on who the donor and the recipient is? So for blood type, it's based on specifically the red blood cells that you have. And red blood cells are just one component of your blood. You also have like the liquidy plasma part, you have platelets, but the blood type is based on your red blood cells, which carry around oxygen in your body. And hopefully, you remember from when we were talking about cells and cell membranes that on the outside of the cell, there are different things that are present that we didn't pay too much attention to when we were actually learning about the cell membrane, but they're going to come into play now. So, we've got these pieces that stick out on the surface of the cell. And that's going to include the surface of your red blood cells. They have these things that are sticking out, and they can be carbohydrates, they can be proteins, they can be combinations of those things. Sometimes it's fats. But basically, there's just these pieces that stick out on the outside of the cell that help cells communicate with each other. And they help cells communicate with other parts of the body, like the immune system. So, when we're thinking about the different options for blood type, what that really comes down to is what sort of molecules are expressed on the outside of your red blood cells. What sort of things do we have sticking out of the outside of a red blood cell? T- uh, Blood type A is going to have certain kinds of molecules that are sticking out on the outside. Here, we're representing it as a pink circle. Blood type B is going to have a different kind of molecule sticking out on the outside of a red blood cell. And here we're illustrating it as blue squares. And type AB is going to have both the red circles that we see with blood type A and the blue squares that we see with blood type B. And finally, type O blood has nothing on the outside of their red blood cells. So here I'm showing you the phenotype for sort of, yeah, what the trait is for these different blood types. So let's take a closer look at the genotype that would lead to each of these traits. And for the genotype, we're going to be using 3 different alleles. So it's one of those traits where we have more than 2 options for our allele types. And those 3 alleles are listed here. We have IA, and it may seem weird to you that we're using I for a red blood cell. It has a reason, but we're not going to get into it because it's not something you need to know. So just take it for what it is that we're using an I for the letter for our blood type alleles. So we have I, and just like with the chinchilla fur, we're using these superscripts to sort of differentiate. So we've got IA, and that leads to this A-type molecule being expressed on the outside of the cell. We've got IB, which leads to the blue squares being expressed on the outside of the cell. And we've got lowercase I, and that leads to no molecules being expressed on the surface of the cell. And you'll see here that instead of saying molecule on the surface of the cell, it's listed as an antigen. So for right now, we're just going to think of an antigen as a molecule that's present on a cell's surface. So this would be an A antigen or a B antigen. And if we have nothing, then we'll say there's no antigen. Okay, so let's use these alleles and start combining them, because remember, each person is going to have 2 alleles for each genotype. So when we're thinking about blood type A, that's the blood type where we get A antigens on the outside of our red blood cells. There are 2 possible options for how we can get this phenotype. One option is to be homozygous for the IA allele, And the other option is to be heterozygous and to have one IA allele and one little I allele. So what this tells us is that IA is dominant over the little I. And remember that little I is when we get nothing present. So if it's a little I, it's going to correspond to no antigens, and that's going to be recessive to any antigen producing allele, such as IA. So both of these options would give us type A blood. Type B blood is going to work really similarly. So we could be homozygous and to have two IB alleles. That would give us the B antigens on the surface of the cell. And then just like IA, this little I is going to be recessive to this antigen producing IB allele. So we could also be heterozygous and have IB and a little I allele, and that would still give us blood type B. And then finally, not finally, second to last, our penultimate blood type is AB type blood. And there's only one option for how you can get this. You have to have one IA allele and one IB allele. And this is going to allow you to produce both the A antigen and the B antigen on the surface of your red blood cells. So if you think back to our beginning slide about the different kinds of inheritance modes, hopefully, maybe you recognize this as being a co-dominant trait. We're sort of getting patches or splotchiness of both the A and the B antigen. They're both expressing themselves in type AB blood. And then finally, our last blood type is O, and this is when you have no antigen. And in order for this to be true, you have to have two of this recessive little I allele. So if this is, we have sort of two modes of inheritance going on here. We have co-dominance going on with IA and IB, but we also have dominant and recessive, sort of like the normal version, because these little I alleles are completely recessive to any other allele. So I don't want to say that I lied to you earlier, but I wasn't giving you the full story. Earlier, we said that antigens are molecules on the surface or the outside of a cell, but specifically, they're molecules on the outside of a cell that are usually going to lead to an immune response. And what I mean by this is that they're a way for your immune system to recognize what belongs inside your body and what doesn't. So foreign particles will have antigens on their outsides. And what I mean by this is things like viruses or things like bacteria. They're going to have these antigens on the outside of the cell that helps your immune system recognize them as not belonging. And then it makes it easier for your immune system to get rid of them. And so if you have allergies, that's an example of your immune system recognizing antigens in things that maybe aren't actually dangerous. So specifically, our immune system should be recognizing things like bacteria, things like viruses. But if you're allergic to pollen, your immune system is getting triggered by antigens on those pollen molecules. So how does this work? The title of this slide is a lot, but we're going to go through it step by step. So antibodies interact with antigens to cause agglutination. There's a lot of alliteration going on here. So remember, we have antigens on the outside of certain kinds of cells. And then we also have these molecules made by our immune system called antibodies. And our antibodies are there to sort of fight against specific antigens. So this is showing a red blood cell, but you could imagine if this was a bacteria that was going to give you food poisoning, your body might produce antibodies that would recognize that this is a bad bacteria that doesn't belong in your body. And then the antibodies sort of attach to the antigens. And you can see that here. We've got the two sort of arms of the antibodies interacting with those antigens. And when this happens, a phenomenon can occur that's called agglutination. And it's a fancy word that comes from like a Latin word that means to glue together. But basically, it just means clumping. Agglutination just means clumping. So the antibodies sort of glue together the cells that they're attached to. And so we get these clumps of cells. And this makes it easier for our immune system to get rid of these cells that our antibodies have recognized. So antibodies, these molecules produced by our immune system, they attach to antigens on the surface of a cell, and then they'll cause this clumping, and it'll make it easier for our immune system to get rid of these cells. because our antibodies should only be recognizing cells that don't belong in our bodies. So maybe you're thinking about this, and it's a little bit concerning, right? Because we just said that types A, type B, and type AB blood all have antigens on the outside of the surface of their cells. So how is this going to work if we've got antibodies floating around that are going to cause our red blood cells to clump up? That doesn't seem like a good situation, does it? The way that our body gets around this is by only producing antibodies to the blood type, to the antigens that we don't have on our blood cells. So if you have type A blood, you only have A antigens present on the outside of your red blood cells. You shouldn't have any of those blue squares. Because of this, the antibodies that are circulating around in your body are only going to be against the blue square antigens. So we'll say anti-B antibodies. And this is fine because you don't have the B antigen on your red blood cell. So these antibodies that are floating around are not going to interact with your red blood cells. They're only going to recognize those B antigens, which you don't have. And then the converse situation is going to be true for people with type B blood. If you have type B blood on your own red blood cells, you only have these blue square B antigens. And so your body is only going to produce anti-A antibodies, the ones that recognize these pinkish circles. But you don't have the pinkish circles, you don't have the the A antigen on your blood cells. So these are just going to float around and they're not going to do anything to your own blood cells. For people that are type AB, they've got both A and B antigens on their cells. So their bodies can't produce either of these antibodies. They can't produce anti-A or anti-B because either one of them would react with the AB blood cells. And then finally, for group O, we've got no antigens on the outside. No antigens present, no circles or squares. So people with type O blood will produce both anti-A and anti-B antibodies. They can recognize the circles and the squares because their red blood cells don't have either. Okay, so we can use this fact to figure out what people's blood type is going to be. And the way that works is with an agglutination test. So when you're looking at these types of tests, what we're looking for is whether the blood sort of agglutinates, whether it clumps up or whether it doesn't. So this is saying it's red blood cells that we're looking at. And here we have a spot of red blood cells on a slide that aren't clumped. It's just smooth looking. Whereas here and here and here, we have pictures of red blood cells that are clumping together. So we can add anti-A and anti-B antibodies to a sample of blood and see whether it clumps up or not in response. And this will allow us to figure out what type of blood we're looking at. So let's take a look at these 4 pictures and figure out what kind of blood is present in each of them. So in this first picture, we're adding, we have 2 samples of blood, a drop here and a drop here, and we're adding anti-A antibody to here and anti-B antibody to here. And we're seeing clumping in both of these pictures. So that's suggesting that we've got an A antigen present and a B antigen present on our blood cells. And this would mean that this blood is type AB. It's got A antigens and B antigens. So when we add anti-A antibody, that's going to react with the A antigen, this pink circle. And when we add anti-B antibody, it's going to react with the blue square right here, and it's going to cause clumping in both situations. So let's look at this next picture. Here, we get no clumping when we add anti-A, but we do get clumping when we add anti-B. So this means that only the anti-B antibody is reacting with our cells. And this means that we have B-type blood with B-type antigens. So our blue square type B antigens are reacting with the anti-B antibody and causing clumping in that situation. But since we don't have any pink circles, there's no clumping with the anti-A antibody. We have the opposite situation here for this third one. Here we get clumping with the anti-A antibody because our type A blood has A antigens, but we get no clumping with anti-B antibody because there are no B antigens present on our cells. And then finally, for this last one, we're getting no clumping regardless of whether we're using A or B antibodies. So that must mean that we have type O blood because it has no antigens present at all. So we'll be using this method to figure out some blood types of some mystery babies, and we'll be figuring out which mothers they belong to. So you'll have a chance to practice this in class soon. So I'll talk to you all later.